All right, so it's 9 o'clock sharp. Hello, everyone. Welcome to our today's Immune Zoom seminar. So it's really my great pleasure to welcome my colleague, Dr. Eric Chang from New York. Actually, presents some really exciting neuroimmune work on our platform. So a very brief introduction for Eric. So Eric currently is associate professor in Feinstein Institute for Medical Research. Eric did complete his undergraduate study in UCLA in neuroscience, and then following pursue his PhD training in New York University, also in neuroscience. Afterwards, he moved to Cornell to pursue his postdoc training in neurophysiology related to a certain disease. So from 2016, so Eric actually started his independent research program in Feinstein Institutes for Medical Research. And then just last year, 2023, Eric was promoted as associate professor in Feinstein Institute for Medical Research. So as I just mentioned at the beginning, Eric is doing really some exciting research in the lab related to the neuroimmune crosstalk. The primary research focus in his lab is on the neuronal regulation of immunity. So his lab actually work across different fields to understand how the nervous and the immune system communicate with one and another. So one of the main focus of his research is to understand how the neuron circuits and the molecular pathways are involved in the sensing and regulation of inflammation within the body. Now it's becoming well established that there is bidirectional communication between the immune and nervous system to maintain the homeostasis and the health. But the precise machinery for these interactions are largely unclear. So obviously, particularly interested in how immune mediators such as cytokines and chemokines are encoded by peripheral sensory neurons in the form of neuronal activity, and whether this neural encoding is disrupted at the onset, progression, or resolution of the inflammation, also a quite important area. So Eric actually tried to advance their understanding of the neuronal circuits that regulate inflammation, which potentially can benefit emerging fields of bioelectronic medicine and then peripheral neural modulation to treat a different kind of inflammatory diseases. So a few different, I think it is really exciting. My lab also are very much interested in neural immune crosstalk. And then there's really a wide space to explore and then to learn new things. So several different focusing Eric's lab, including selective activation of vagal fiber subpopulation for regulating immune responses. And then they also try to develop bioengineering devices and the method for interfacing with the peripheral nervous system. And then they also try to understand the neural physiology of neuronal circuits in disease conditions, also the molecular and structure correlates of synaptic functions. So with a really very fruitful research outcome, Erica got numerous awards and honors, which I'm not going to list them all, but particularly including the Helmsley Scholarship for Interdisciplinary Research, and then BBRF Young Investigator guarantees. So I think without further ado, Eric, thank you so much for presenting your work, share your work with us today, and then please go ahead.
Okay, thank you, Chuan, for the nice introduction. Good morning to everyone here on the East Coast in the US, and good evening to those of you joining from China and other parts of Asia. Again, thank you so much for the invitation, and nice to see you again, Chuan. This is a really nice, Immune Zoom is a very nice platform We appreciate the opportunity to share the work on, like you said, trying to figure out how the nervous system, in particular peripheral nervous system, as well as a little bit of work in the brain itself and the CNS, is involved in the regulation and encoding of innate immunity and inflammation. So first, let's see. So first, who we are and where we are, because since you were interested, and we get this question often. So I'm speaking to you from the Feinstein Institutes in New York. We're actually located in Long Island, and I work very closely with a group of faculty members here, including Dr. Kevin Tracy, who's the president of the Feinstein Institutes, as well as other faculty members and a group, a larger lab group that you'll see some of these names as I go through the work. But we are part of a large health system in New York called Northwell Health, which is massive. It's actually the largest private employer in the state of New York. So there's 85,000 plus employees. As you can see on this map, there's 23 or more hospitals. We just, I think, acquired another health system last month. And the Feinstein Institutes, which is where I'm speaking to you from, is the research arm of this a very large health system. So we're located, as I said, in Long Island, just outside, about 40 minutes east of Manhattan, and just outside of New York City. And so on this other map, you can kind of see a little more where we are with this green spot here. Manhattan is here. The Long Island is long because it's called Long Island because you see it's a Long Island. But if you zoom out on this map, actually, some of you may be aware that there was a solar eclipse that passed through North America and parts of the US this past Monday. So just on April 8th, there was a part of New York. We weren't in the so-called path of totality, where the sun was completely blocked by the moon and it'd be completely dark, but we were very close to it. We're only a few hours north of it, a few hours south of it, because we're down here. We saw, this is just earlier this Monday, we had a bunch of scientists go outside at that time, looking up at the sky, some people going out with their lab coats, probably some kind of violation. And these are some pictures from my, poor pictures from my camera phone through the protective goggles or glasses. And then, of course, you can see if you look at the Google Trends search after that, people search for "my eyes hurt." Why do my eyes hurt? Right after the solar eclipse. So despite many scientists and news outlets and everyone saying don't stare directly at the sun, many people probably stare directly at the sun. So that was, I think, a pretty cool thing that happened just earlier this Monday. The next solar eclipse that is visible in the US is going to be in 20 years. But for New Yorkers, it'll be not until 2079. So it's a pretty rare event, and got, I think, a lot of people interested in science generally. So just a little bit of geography and some astronomy, I guess, to start before we go into neuroimmunology, which, as Xuan mentioned, is something that we work on, particularly the crosstalk and interactions between the nervous system, both in the brain and CNS, as well as within the peripheral nervous system, or the PNS, and how they communicate with different compartments and different immune cells of the immune system. Mostly focused, but although not exclusively, on innate immunity. So, as this audience is likely aware, the crosstalk between different parts of the nervous system and immune system is a growing field that's really kind of, I would say, expanded tremendously in the last decade. There's potential for crosstalk and communication between immune cells and nerves and different peripheral ganglia that I'll tell you about. And what we're particularly focused on is how, and I'll show you some results about peripheral nerves, particularly somatosensory and viscerosensory communicating nerves, talk to immune cells and might be able to encode, detect and encode different immune mediators. We have a focus on cytokines, but obviously different chemokines, DAMPs. We have some work in HMGB1 that I'm not going to show you here today, but how are these immune mediators when they're released, at perhaps a local site of inflammation, how are those detected? Or are they detected by nerves, and if so, how? And how is that encoded in the form of neural signals? So this nerve here in this review from Isaac Chu and Cliff Wolf is a peripheral nerve. It could be the vagus nerve, which is one of the nerves we study intensely. It's cranial nerve 10. And the reason why we study it is, as you can see, neuroanatomically, it's a cranial nerve, so it descends from the brain. It's actually a pair of nerves that descends from the brain stem, and you can see innervates all our major viscera. So, and it doesn't do that in a straight line path. It takes a wandering path, which is what vagus means in Latin. Vagus is Latin for wandering. And you can see it's, as many of you know, is part of the autonomic nervous system. And it controls, many major vital functions that are critical for our survival: breathing, heart rate, blood pressure, glucose levels, hunger, gut motility, and importantly, for some of the work I'll tell you about, autonomic reflexes that again are these are not things that we're consciously aware of. These are just things that are autonomic, part of our autonomic nervous system. But I think what we're trying to figure out is what are the different molecular mechanisms that mediate some of these vital physiological functions, and how does the neuroimmune communication play a role in that? And what's interesting is that actually the majority of fibers in the vagus nerve, in both in preclinical models and in humans, about up to 90% of fibers actually sensory afferents, carrying information from the body up to the brain. So that's, I'll tell you a little bit more about that. But some of you may have heard of vagus nerve stimulation, or VNS, which is essentially putting a cuff electrode to cuff typically the left vagus nerve at the cervical level. And this is FDA approved here in the US for a long time to treat medication refractory or treatment resistant epilepsy, as well as depression. So these are typically patients that don't respond to first line treatments, which are typically drugs for a couple rounds. And then just a few years ago, VNS has been approved for motor recovery following stroke. And there's now a company here based out of California that is in late stage clinical trials to testing VNS in patients with rheumatoid arthritis. So I think what's coming soon on the horizon, potentially very soon, is that VNS could be approved. This is, a VNS implant, which is the size of the fish oil pill, onto the left cervical vagus of humans. So patients with chronic inflammatory disorders could have this as an alternative treatment potentially soon for inflammatory disorders. And that indication came out of work from Dr. Tracy's lab here at the Feinstein Institutes 24 years ago now. But this was work in a rat model of endotoxemia, basically showing that if you electrically stimulate the vagus nerve, this is in a rat model, at the cervical level, you get a reduction in circulating cytokines. So this is a paper published in Nature, but injecting LPS, lipopolysaccharide, which is a bacterial endotoxin, increases levels of serum circulating tumor necrosis factor, or TNF. And then when you perform a vagotomy, which is what VGX is, cutting the vagus nerve, it's kind of like releasing the brakes, and you actually get more inflammation. However, you perform a vagotomy but stimulate the end still connected down into the viscera, into the end organs, you get this massive reduction in levels of TNF. So this was kind of a first critical demonstration that electrical activity on the vagus nerve at the cervical level could dramatically reduce levels of circulating cytokines to protect, in this model, against lethality and improve survival. And then work from our lab and others over the decades since have kind of delineated a circuit that I'll tell you a little bit more about in this last paper that is termed the inflammatory reflex, the vagus nerve mediated inflammatory reflex. And in this recent paper, well, not that recent, 4 years ago, this work was led by Adam Kressel, who is a surgeon getting his PhD in the lab in a program we called the Elmezi program. And what Adam did was he wanted to show where are the cholinergic neurons that originate the vagus nerve efferents, shown in green in the schematic, that descend in the vagus nerve that were presumed to mediate this anti-inflammatory action. And so first what Adam did was injected AAV into the dorsal motor nucleus of the brainstem, and then also inject HSV retrograde with M-Cherry tag into the spleen. And what he found was that they converge, he hypothesized that they would converge in the celiac ganglion. And so these are images. This paper's published, so I'm not going to go spend too much time on this. But basically, if you look in the celiac ganglion on this last merged image, the red neurons are noradrenergic neurons that were retrogradely traced with HSV1 with M-Cherry. And the green little squiggles are actually terminals from the AAV labeling into the brainstem. So they co-localize with synaptophysin. So this is basically a cholinergic, parasympathetic, direct interaction with noradrenergic neurons, canonically sympathetic, going into the spleen. And then functionally, then you wanted to test using optogenetics. So this is in channel rhodopsin animals, again, targeting the dorsal motor nucleus, the DMN, which is where the origin of these cholinergic efferents ultimately signal to the spleen originate. And so if you look in the brainstem in the DMN, you can see, just to verify that the cholinergic neurons have the channel rhodopsin with YFP. And then he put a fiber optic cannula there in the DMN stimulated. And then to see whether or not that reduced TNF, and with yellow light stimulation, which doesn't activate channel rhodopsin, you do not get the reduction in TNF. This is with LPS, acute endotoxin model. But when you use blue light, which activates channel rhodopsin, you get, again, this decrease in levels of circulating TNF. And then because Adam was a surgeon, he did this pretty difficult experiment, which is to put a very small nerve cuff electrode on the mouse splenic nerve, which is a tiny nerve. And then, so put a nerve cuff electrode on the splenic nerve, record, action potential neuroactivity there. When you turn the light on in non-channel rhodopsin control litter mice, the activity doesn't change. However, in channel rhodopsin animals, when you turn on the blue light, you saw an increase in neural activity of the splenic nerve. So, this kind of confirmed that the DMN is the source of this activity on the cholinergic efferents that travel down the vagus nerve, synapse onto the celiac ganglion, noradrenergic neurons, ultimately going to the spleen. That work, in addition to many other papers from the lab, have kind of delineated this sometimes called a cholinergic anti-inflammatory pathway of the inflammatory reflex, which is one example, I think a strong example of neuroregulation of innate immunity through neural activity on the vagus nerve. So we know in the brainstem, this is obviously a human brain, but in the DMN of the human brain or mouse brain that we showed, we have electrical potentials traveling down the nerve. They go to the celiac ganglion and then activate splenic nerve, which is actually noradrenergic, releasing norepinephrine onto CHAD-positive T cells in the spleen that then release acetylcholine that binds to alpha-7 nicotinic receptors, and that reduces cytokine production and release. This is, like I said, an important pathway for regulating inflammation. And the reason why it's important and now potentially could be the basic science mechanism for a clinical therapeutic treatment in patients is, as many of you are aware, the treatments for many of these inflammatory disorders, let's say rheumatoid arthritis or Crohn's or psoriasis, are monoclonal antibodies, of which here in the US you see commercials of all the time by these drug companies, direct-to-consumer commercials. And this is a multi-billion-dollar industry Humira and Buril. These are, monoclonal antibodies that are, given IV or injected. They are expensive, and they also, they work in some patients, but they also, don't have effects on symptoms in many other patients, or the side effects for some of these monoclonal antibodies are too severe for some patients. So, if we're able to intervene, Juan mentioned and I won't go into this, but bioelectronic medicine is one approach, which is instead of using biologics like this or traditional pharmaceuticals, if you understand a neural circuit, you can intervene by modulating that circuit. In the case of VNS implants, that are being implanted in humans now, for epilepsy and depression, this has already been implanted in hundreds of thousands of patients. So it has a very positive safety profile, and there are very few side effects. But once we understand how cytokine inhibiting neural potentials along the vagus nerve and other peripheral nerves might control different aspects of inflammation, that's when we can start to work with bioengineers and material scientists to design devices, which we do some of that as well, to intervene and interface with some of these nerves to treat disorders. So, that's the what's called the motor efferent arc of the inflammatory reflex, which, as I mentioned in an earlier slide, is really only a small percentage, a minority of fibers on this nerve. The sensory afferent arc, as we all learned in our textbooks, a reflex, a neural reflex, has a sensory afferent input as well as a motor efferent output. And a lot of what we're doing more recently in the lab is trying to figure out how does the sensory arm of this arc of this reflex circuit work, and do sensory neurons in the periphery, including vagal sensory neurons, detect different neuropeptides, chemokines, cytokines that are in either end organs or in different tissue sites? How do they detect them and then encode that information to send to the brain? Obviously, I think, well, many neuroscientists are familiar with this, but the French philosopher and scientist René Descartes, almost 400 years ago, delineated a pathway for the sensory part of a reflex circuit. You need a stimulus. In this case, it could be a cytokine, a receptor. We know, I won't show you, but we have immunohistochemistry showing cytokine receptors are on vagal sensory neurons. The pathway is the vagus nerve, and then the brain is involved to integrate and then trigger, in this case, in this picture from Descartes, withdrawal from the flame. In our case, it would be a cholinergic output originating from the DMN to reduce cytokines. Our work, so our lab has done some work, this is a work I'll show you with vagus nerve recording. So this is putting, again, one of these cuff electrodes. You can see it here. This is a commercial cuff, neural electrode cuff from Cortec. You can't really see it here because the nerve is translucent, but in the mouse, there's a vagus nerve along this cuff. We connect that to an amplifier, and then we can record basically, what are called compound action potentials, because these aren't single fiber recordings. And the vagus nerve, even in a mouse, is about 3,000 to 5,000 fibers per side. In the human, it's about 100,000. So given the connectivity and traffic going up and down this nerve, there's a lot of neural activity, as you can imagine. In this model, this is done under anesthesia. But basically, when you inject saline, you don't really get a change. However, you inject TNF, this is injections IP, you get, over the course of minutes, a change in the level of neural activity. And similarly, with another pro-inflammatory cytokine, IL-1, it seems like you get an increase in neural activity over the course of 10 minutes. And again, this is an IP injection, so this change in vagus nerve activity could be through potentially a humoral route. And to follow up on that, we work with a lab here, Theo Zanos's lab here at the Feinstein Institutes. There's another faculty member, and his lab is a machine learning and data analytics lab. And again, using vagus nerve recordings, what he did was record from the vagus nerve, and during IP injections of TNF or IL-1, what their lab did was use different clustering and machine learning techniques to identify compound action potential clusters, shown here, that appear in response to different IP injections of these cytokines. So, and then what they did is developed a decoder, a naive Bayes decoder, to see if you just track this vagus nerve activity alone, how well does the decoder do to know that a saline was injected or IL-1 or TNF was injected? And it turns out the performance of the decoder was about 83%, which is pretty good. And again, points to the fact that there is cytokine-specific activity that you can record with a cuff electrode on the vagus nerve. And then last year, we published some work identifying a subset of TRP channels, so TRPA1, which is involved in inflammation, as many of you likely know. TRPA1 is transient receptor potential anchorin 1, and a subset of fibers on the vagus nerve that express TRPA1 appear to mediate IL-1 induced hypothermia, as well as being able to activate aspects of the inflammatory reflex. So in this work led by Harold Silverman, who was a graduate student in the lab, what Harold did was, again, do these vagus nerve recordings. So these aren't individual neural recordings or individual fiber recordings. These are cuffing the whole vagus nerve of the mouse at the cervical level. And following IL-1 injection, again, you get an increase in activity. In SYNCRE TRPA1 flox animals, where TRPA1 is removed from neurons specifically, you do not get that increase. And then then there's a compound that not many people know of, but Harold found was called optovin, which is a form of optopharmacology. So you put optovin onto the nerve or in a part of the brain, and it binds to TRPA1 channels and makes them light sensitive. So it's kind of a variant of optogenetics. It's called optopharmacology. And when he did that, again, in this LPS model of acute endotoxemia, you shine light in wild-type animals with optovin, you get this decrease in TNF. In TRPA1 knockout animals with optovin, you do not get that decrease in TNF. And then finally, in this paper, which was published last year, he performed chemogenetic experiments with GQ dreads in TRPA1 pre-mice and showed that activating this set of channels with CNO does, again, increase levels of circulating cytokines. So, together, this set of work is identifying that the vagus nerve is certainly involved in this, and we're recording directly activity on the nerve, and this, the involvement and implication of the vagus nerve in mediating, for example, IL-1 signals or TNF signals has been known since the late 90s from work from Linda Watkins and colleagues that did vagotomies at different levels and showed that basically that you could block the fever by cutting the vagus nerve. So the vagus nerve has long been known to be involved, but now we're starting to see what are the actual signals on the nerve. And then in more recent work, so at that point we knew that cytokine signals are carried by the vagus nerve, but we want to know, do individual neurons actually, how do they, or first of all, do they, and how do they, if so, transmit those signals? So the vagus nerve, as you've seen now in several diagrams, innervates our major viscera. And when there's injury or infection, obviously there's the innate immune response involves inflammation, and cytokine release is part of that initial response. And whenever there's increased inflammation in any of these end organs, you also have the expectation that cytokines are elevated. So does the peripheral nervous system, which is in the vagal ganglia, the nodose ganglion, is where these vagal sensory neurons that reside, and then the next synapse is actually in the brainstem, the nucleus tractus solitarius, NTS, which actually neighbors the DMN. So you can kind of see the buildup of a sensory circuit going to the NTS, then communicating to the DMN, and sending that motor efferent arc to regulate cytokine release. So this is another paper we published last year, basically developing a set of tools and techniques for imaging this nodose ganglion, which is again where the sensory neuron cell bodies reside of the sensory afferent vagus nerve. So you can see where it is in the mouse. It's kind of in this very tricky spot because it's at the base of the skull, very deep in the neck. So this was work led by Tomas Huerta, who's a graduate student in the lab and now a postdoc. And he developed a surgical technique and approach for isolating the vagus nerve. And this is now in this picture here, looking down next to the trachea. So it's like around here, and you can't really see, but there's a bulb at the bottom of this, which is the nodose ganglion, the jugular nodose ganglion. And what he does is actually cut it free from the brain, but it's still connected to the viscera and the rest of the organs, and put it on a platform. We use something called a miniature microscope or a miniscope to image this in V-glut GCAMP6 mice. And then what we're doing is applying different agonists in this paper to the vagus nerve itself. So this is different from the prior work, which is mostly IP injections, which, obviously could have some neural communication, but also has obviously the humoral route, which we know a lot about, but it's obviously slower than a neural route. And so again, these are, in this paper, we're using V-glut2 GCAMP6 mice, GCAMP6F, and imaging with a miniscope. And we use a Python-based analysis tool called Cayman, calcium imaging analysis, with some modifications, because this miniscope is actually developed from a group from UCLA, where I went to undergrad, and it's kind of an open source, open science project. But it was actually designed to image brain targets with a grin lens. So we're using it in this kind of unusual way to image this vagal ganglia that house the sensory neurons of the sensory vagus. So we had to make some changes to the code to be able to extract the signals and understand them in a way. So, Cayman allows us to extract spatial ROIs, which are the neurons in the nodose, as well as plot calcium transients as traces. This is what it looks like when you put capsaicin, for example. This is a video of the calcium transients in the nodose ganglion when you put capsaicin onto the vagus nerve. So, again, you can extract the spatial ROIs, you can plot them. We developed some Python-based tools to select out, non-selective responders, remove artifacts. This paper's published, so I'm not going to go too much into it. But then what we did for this was TRPA1, which I already mentioned earlier, can be activated by AITC and other different stimuli. TRPV1, probably many have heard of, which is the channel for capsaicin, which is the hot element in chili peppers, and glutamate, who are the three agonists we use. And using our analysis tools, we're able to extract different features, we call them, the peak amplitude of these transients, integrals, durations, number of peaks. And what we found was that the features of these calcium transients differed for the different classes of agonists that were applied to the vagus nerve. So imagine that the vagus nerve is innervating an end organ, whether your organ of interest is the lung or the gut or the liver, and different stimuli are coming to the terminals. It appears that there are different patterns for the neural encoding of those stimuli. And we tested that with putting aspects of those features, in this case, amplitude, duration, peaks, and integral, into a random forest model classifier to see, just having the neural information alone, how well does the model do in predicting what the vagus nerve saw, essentially. So, typically these models require, they do very well with hundreds of thousands or more of instances and data and training points. So, in this case, we had hundreds of traces, but less than 1,000. So the data sample was low for this type of model, but it still did fairly well. You can see the accuracy 71, 78, 82 for different classes of agonists. And extracting what are called the SHAP values from this, we know different aspects of these calcium transients, in particular the amplitude and duration, seem to be the most important features that would help the model to in its accuracy for knowing what the vagus nerve saw. So, what we took away from this is that this information is already being encoded at the level of the periphery before it gets to the NTS and into central structures, which we find interesting. And what we want to know really is, is a similar type of transformation or encoding happening in regards to cytokines. So this is, again, focusing on the nodose ganglion. We have the same imaging setup in VGluGCAM6 mice because all the sensory afferent neurons are glutamatergic. So this is, again, the setup under anesthesia. And then this is kind of the raw image of what a nodose looks like. This is actually the delta F over F, the calcium transients with TNF applied to the vagus nerve, and then seeing neurons that directly respond to TNF. And so, and this is an example of, again, imaging in the nodose ganglion, and we can see that the interleukin-1 beta, or IL-1, responding neurons actually have a very different type of signal. They're low in amplitude and they tend to be burstier compared to TNF. And if you remember back to the capsaicin video, actually the dynamics of these cytokine-specific neural responses in the nodose differ pretty dramatically from capsaicin and AITC and glutamate. So this is in this work that is unpublished, we're hoping to wrap up soon. So this is not finalized, but we've looked at TNF, IL-1, and IL-10, canonical anti-inflammatory cytokine, and you can kind of see at a qualitative level here that the dynamics and kinetics of the different responses of these neurons to these individual cytokines differs. We can quantitate that. And again, this is not finalized data in progress. This is work led by Tomas Huerta in the lab. And basically, it appears that there is cytokine-specific neural encoding at the level of the nodose before we get into the brain, and that IL-1 has a particular type of calcium transient response. TNF has a different one. IL-10 has its own as well. And you can look at them together and see that they vary amongst these different features that I spoke about. But what's also very interesting is that if you look, some labels are missing here, but basically, if you look at when a group of neurons responds to IL-1, you can see this, the first bin, these are about 3 to 5 second bins. So you can see that the responses happen rapidly. And this indicates that these neurons are responding in a rapid fashion because we're putting them directly on the vagus nerve. And we have immunohistochemistry. I'm not showing you that there's cytokine receptors on the nerve as well as on the cell bodies. But what's also interesting is that there are some neurons that respond non-specifically to both cytokines, TNF and IL-1. And there are other neurons, as we know, because the vagus nerve is involved in mediating so many signals going up to the brain that have nothing to do with inflammation or cytokines. There are other neurons that don't care about cytokines and are responding to other things. For example, the lung status of having to do with respiration. This is a work in progress, but we're very excited about showing these nodose ganglion neurons that encode first of all, they directly respond to cytokine application and they encode them in a unique way. And then another thing we're looking at is how does that signal or does that signal change when there's inflammation in the context of inflammation? So in this model, we're using DSS colitis, which is dextrin sodium sulfate added to the drinking water of mice. And basically, we can rapidly, within 6 or 7 days, get inflammation of the colon. And what we wanted to see is how do those neural responses that I showed you change, or do they change because there's colonic inflammation and elevation of serum cytokines. So there's, not showing you here, but there's elevation of cytokines in the tissue as well as in circulating levels in the serum. In the model, like I said, you get reduction in body weight, disease activity score goes up. The peak is about 7 or 8, which is where we do calcium imaging. And then I'll breeze through this, but basically their colons are shortened in the DSS mice, and there's histological abnormalities in the gut epithelium. And then what we find so far that's interesting is that it appears that there's reduced amplitude at the baseline condition. So these aren't cytokine specific responses I'm showing you here, but I have that, and we're preparing that for submission. But what we're finding is that there is definitely a decrease in baseline activity of these responses, the amplitude of these responses. So we develop a neuroactivity index. You can see in the context of inflammation, so this is just DSS colitis, colonic inflammation, but like I said, serum levels of cytokines are also elevated, that you get this neural change. You know, we want to know, first of all, what does that mean? And how does that affect, you know, the neural responses to cytokines that I showed you earlier? You know, so I think what we know so far through work in our lab and some others is that information about cytokines in the body, in the periphery, are being encoded, are being detected and encoded by vagal ganglia, specifically nodose ganglion, in the periphery prior to entering the central nervous system. And you know, if we compare this to other modalities and strategies and that have developed over, you know, through evolution in other sensory systems that you know, systems neuroscientists are familiar with, you know, for encoding taste, you know, work through you know, Charles Zucker and other labs has shown, you know, that there's a labeled line system that, you know, the stimuli comes in at taste buds and then follows a labeled line into the area of the brain. Other sensory modalities that we know, you know, use different types of coding systems. They could be temporal codes responding to a stimulus, or a rate code encoding a particular aspect of the stimulus. And what we want to know, which I don't have the answer to yet, but we're very interested in, is there a similar, what are the rules, if there are any rules, but presumably there are rules for the population coding of immune mediators within vagal ganglia, such as the nodose ganglion. And you know, what we know is that there is cytokine-specific neural responses at that level. And you know, this idea of using some of these tools from neuroscience that you know, including voltage indicators, moving beyond calcium indicators, is something very interested in doing, because it appears that you know, there's a new kind of framework or understanding of how the immune system releases different inflammatory mediators in this case, and how the nervous system encodes that information, right? And I think we're really only at the beginning of understanding these types of first principles. So you know, obviously, we are familiar with the 5 senses of, you know, sight, touch, hearing, taste, and smell. These are the 5, you know, exteroception, it's called. And you know, some people call the 6th sense proprioception, which is you know, just the sense of where our body is, where our limbs are. And then, you know, these classes of interoception, including potentially cytokine detection and signaling of cytokines and different immune mediators to the brain via the periphery, is kind of a new exciting field that we're hoping to make some discoveries in. And you know, what we want to know is, what are the first principles for neural encoding of these cytokines? Can we discover patterns to understand this? And then more importantly, can we use that understanding to develop better treatments for inflammatory disorders, or better understand the different neural circuits involved in mediating some type of inflammation? Use that, going back to the bioelectronic medicine, to design and interface with design devices to interface with nerves and modulate that inflammatory signaling going up to the brain. And you know what, they don't have time to cover, but work from many other labs in you know, using transcriptomics. This is, you know, has shown, not surprisingly, that these vagal sensory neurons, you know, I won't go into this, but you know, there's jugular and nodose. The jugular are more somatosensory-like, more DRG-like, dorsal root ganglion, and they send somatosensory signals, and they derive from the neural crest. Whereas the nodose are placo-derived, and they as I mentioned already, innervate the viscera. So you know, there are many different clusters genetically of these different vagal ganglia. And like I said, work from Steve Lieberle's lab at Harvard, Roy Chang's lab at Yale, has really shown that these are incredibly complex and polymodal population of neurons is very heterogeneous. So and we're just at the beginning of understanding what they're doing. Obviously, our lab is very interested in what they're doing from the sense of immune signaling, but many other labs are interested in, its role in gut motility or sensing different glucose in the gut, or what it's doing in breathing, what it's doing as far as modulating heart rate. This is a very interesting field in this falls in the space of interoception, but that we're starting to understand. And of course, there's some work in our lab working on the brain targets as well, that I'm not going to talk about, but and there's work from other labs showing that if you induce inflammation, or presumably if there's chronic inflammation even in a patient, that is encoded in cortical neurons of the brain, right? So this is work from Ozzy Roll's lab showing that insular cortex neurons encode aspects of peritonitis or colitis, like we studied. And then that later on, you can reactivate that cortical population and recapitulate some of the inflammation. So, this and some other work, there's work from Jeff Friedman's lab showing areas involved in sickness behavior. So, using technologies like, calcium imaging, trap-based imaging, chemogenetics for reactivation, a lot of labs are starting to show that obviously there are central nervous system brain regions that actually respond to cytokines and different types of inflammatory events. I think I showed you a little bit about potentially the vagal sensory neuron route that might take inflammatory signals from the body and the periphery up into different parts of the brain. So, we're very excited about that type of work. And I'll just close on this slide, which is, I hope I've shown you, convinced you that the vagus nerve mediates many aspects of physiology that are vital to our survival, and also aspects of regulating immunity and also sensing aspects of inflammation. And, this is the vagus nerve, the reason why I think it's, so interesting to many potentially different labs that work in different diseases and, works on different physiological functions or end organs, is that because the vagus nerve innervates all these major organs, there are many different compartments you can kind of build a career on, really. And I didn't mention this work, but we also published work showing that if you target innervation of the liver at the porta hepatis with focused ultrasound, you can change metabolic outcomes in a Western diet model of obesity. But there are many different groups starting to show and delineate what are the different molecular mechanisms that mediate different functions in the gut or the pancreas or the stomach or the liver or the lungs. And some of these reflexes are have been known for over 100 years. So we knew what they do, to protect, when something goes down the wrong way and you have to cough. Now we know that piezo2s are the ones that first sense that, and or trip channels are the ones that mediate that. And using the tools like chemogenetics, optogenetics, and different types of newer imaging, we can start to really identify, in this case, what are the vagal sensory neurons that are involved in this reflex. And we're interested in the inflammatory reflex, obviously, but other vital reflexes that the vagus nerve is also implicated in are also obviously interesting to us. So, finally, I just want to thank you to everyone. Thank you to Chuan for the invitation, for listening. These are the people I work with here at the Feinstein Institutes in Long Island. And this is the group in the lab that I work, that works closely with me, as well as a larger lab group. And want to acknowledge, of course, our funding sources as well. And thank you, and happy to take any questions.